the last in the session. Maybe I will share about the potential differentiation of mesenchymal in hypoxic needs for retinal disorder. Maybe this is a simple presentation. As we know, there is a chief area for personalized medicine. One of that is cell engineer. And there is our center in Surabaya. There is collaboration 3 institutions, School of Medicine, Dr. Stone Hospital, and Laboratory Stem Cell. And there is our roadmap in the clinical trial. We make a focus on safety, efficacy, and side effect. And there is our continuing of the roadmap and becoming clinical trial in the file disease. And there is our center in Indonesia, Surabaya. And there is a beginning in the 1990 with Punbeng at Dr. Stone Hospital. And as we know, the stem cell have a basic nature of stem cell with plasticity and self-renewal ability to differentiate it and proliferate it. So with the promising of stem cell, one of that is to treating of degenerative disease in here, like diabetes, myocard, stroke, and also maybe Alzheimer. And approximately, as we know, United States alone, approximately 128 million individuals would benefit from stem cell treatment. So MSC itself, there is cause of multipotent differentiated capacity, so the tropic with a potent immunosuppressive effect and ability to the vascularization, and the utility to treat a large variety of pathologies, including retina disorder. So if we talk about the retina, so it's very important we know about the embryology of retina. From the hierarchy of cell, so retina is developed from neuroectoderm of the prosencephalon. So this encephalon become optic groove, optic vehicle, optic cups, and becoming the retina, optic nerve, ciliary body, and iris. So the anatomy of the retina should be coming to many layers. In here, so we focus in the disorder and retinitis pigmentosa, as we know, is might be related genetic disease. So usually, generically, I describe progressive cause of visual loss, which attribute to loss of fiber of photoreceptor, especially root and cone photoreceptor, and associated also pigmentary changes in the retinal pigment epithelium. So will the patient have a visual loss. So the photoreceptor that predominantly the effective might be root and cone photoreceptor and all the epithelium cell. And given there is a primary root and cone dystrophia, so the patient with central vision is separated lesion and loss of the vision. So there is maybe anatomical root cone photoreceptor and the pathway of the light perception in the patient. And there is retinitis pigmentosa with pigmented of the eye. So with analysis based on that, based off embryologies of retina from ectoderm, so need stem cell can develop to 3 germ-layer. Usually, mesenchymal stem cell is developed only 2 germ-layer, ectoderm and endoderm. But because the embryogenesis of retina from ectoderm, so we need to manipulate change mesenchymal stem cell to pluripotent, which needs manipulation. Stem cell manipulation, so stem cell have passed through 2 conditions before transplantation. The first from isolation in vitro condition, and the second in vitro manipulation when the cell before transplantation. Usually we can manipulate the cell in vitro environment. Some of kind is with hypoxia manipulation. So as we know, a stem cell needs can determine by compartment composed of cellular and acellular engraftment. So become the control of rate of stem cell proliferation and determine the fate of stem cell ductor and also protect the stem cell from exhaustion and death. So in here, to focusing, usually when to higher concentration, so approximately 5 or to 10 times more the concentration of O2 in the natural niche from organ in the milieu in the body. So as we know, in the body, so usually the oxygen concentration for lung parenchymal is need 4 to 14% as circulation, liver and kidney and also heart. So in the brain, usually less of oxygen concentration, 0.5 to X% as we know of eye, bone marrow and adipose tissue. So there is regulation transcription for hypoxia condition, so make HPH hydroxylase increasing. So the regulation transcription for HEF1 alpha deposit and translate for gen to express glucose transporter and also HD8, PKD1, and chemokine coreceptor 7 and 4, as we know, is important for metabolism, as we know, it's important for migration, and as we know from angiogenesis. And also the suppression of mitochondria, so lack of oxygen make a decreasing of ROS, and there is enlargement of telomerase, and also DNA demate protection, and also inhibit of senescence and aging. There is for migration. So hypoxia condition make maintain proliferation, plasticity, genetic stability, and increasing of enhancement of mesenchymal stem cell. In here, our study, so we take mesenchymal stem cell isolation from bone marrow, and we culturing, and we take for passage 1 until 3rd, and we become a single cell before ejected. And we make a niche with comparison normoxia and hypoxia. From GFU, normoxia is less more than hypoxia condition. And also expression for OX4 and SOC2, the expression from hypoxia in 3% concentration of oxygen. So there is an expression with immunocytochemistry. And there is a normoxian, no expression OX, OX4, and SOX2. And also, as we know, P63 expression is becoming in the family of P53. There is a normoxia, no expression, and hypoxia 3%, there is expression P63. So after that, we preparation for mesenchymal stem cell. And before injected, we validation with a pluripotency, contamination, and purity. And then we collaborated with eye clinician ophthalmologist. So after single cell, separate cell, so become under microscope, we injected to intraocular, intravitreal. And before that, we analyze from animal, so there is develop of retinal cell in the layer in animal eye. So the evaluation 3 months after injection, so 3 patients with retinopathy diabetic with anamnesis blue revision, eye pain, and visual F5/10 with autologus mesenchymal stem cell injected intravitreal. So we injected twice with a range 1 month. So from analysis become a brightness, decrease of pain, and the visuals become 5/7.5. And 3 patients with retinopathic diabetic also with blue revision also and cannot read and cannot driving. With eye pain also, the visuals is 1/60 with autologus mesenchymal stem cell intravitreal. With once injection, so decrease of pain, can read, short messenger, and can driving, and also the visuals become 5/20. And 3 patients with retinitis pigmentosa with light perception only. So with Autologus mesenchymal intravitreal for 3 times, they can read such a messenger and converts the television, and the visuals become 3 per 60. But one of the patient left eye post injection twice, the patient find bubble in the vitreous, so we done a vitrectomy for the patient. So beside that, we also a clinical trial to many cases from August 2014. We have 130 cases with many wild of many diseases in our department. So when it's a sort of stem cell, usually we use autologus and less than allogenic stem cell, and we use a mesenchymal, hematopoietic, NK cell, and nerve cell of neuron cell. So there is our center in Surabaya. Beautiful of Indonesia may be as beautiful as shown in on site. Thank you. Yes.
I'm going to ask you about the improvement in the sight. It was quite marked. And the improvement that you got in sight was really quite good, quite marked. And that was quite a big jump, I thought, from 5 to 7.5 and things like that. So the patients must be very pleased with that sort of response.
Yes, the improvement is after 6-month injection. So we done not only one-time injection, but booster through until 3 times. With rinse for booster is 1 month.
Okay.
Yes, but some patient maybe because I don't know, because retinitis pigmentosa itself about the disease, so the vitreous is become not so clear. So we done a vitrectomy for the patient. After vitrectomy, the patient can visual with watching TV. Yes.
But they could all see in the end. I mean, the ones you showed us were all able to see with improvement.
Yes, improvement. All of improvement.
Mm.
Yes.
Okay, thank you.
Thank you.